everybody, and welcome to OSU Extension Agronomic Crops Team's first ever virtual corn college and soybean school. I'm Mary Griffith, Extension educator in Madison County, Ohio, and I'm one of the agronomic crops team leaders along with Amanda Doritas and Laura Lindsay, and the three of us will be moderating throughout the day. So thank you for joining us.
All right. Well, thanks for joining for the second half of our program. We're going to turn our attention now to soybean. So I'm Laura Lindsay. I'm the soybean and small grain extension specialist here at Ohio State. This afternoon, we have several soybean specialists, including Dr. Mark Laux, who will be talking about weed management in soybean, Dr. Ann Dorrance, the soybean pathologist, and Dr. Kelly Tillman, who is going to focus on insect management in soybean.
Will be about the results from the Soybean Cyst Nematode survey that we've been doing for the last couple of years, and then also seed treatments. Really kind of what the status and a little bit of a heads up for this year with some of the diseases we're already seeing. So with that, Soybean Cyst Nematode, there's been a big national survey that we've been participating in, funded through United Soybean Board, the North Central Soybean Research Program, and of course our own Ohio Soybean Council. But the theme is what's your number? Take the test, beat the pest. And so we've been participating in this to enhance the educational awareness of soybean cyst nematode in Ohio. It's helped us with some sampling. And it's also helped us to understand how the resistance is working that's been deployed for over 20 years. So with this, for symptoms during this season, there really aren't a lot. I mean, this is kind of a wimpy pathogen if you ask me. So, here are the females, the cyst females. You can see them lining up just like they're in a cafeteria line here. Here's a rhizobium nodule for size. But you can find these usually in the midsummer, later in the year when the numbers start really increasing. And just dig up the roots, kind of shake them off, and then slowly these will glisten because they're very moist compared to the sand granules. We manage cyst nematode based on population numbers. So with the population numbers, you've got 0 to 40 eggs per cup of soil, continue to monitor the field. 40 to 200, that's a trace level. But when we start reaching these levels, such as low, we can begin to see some yield loss on a susceptible variety. This is when we recommend switching to resistance. But when we're up into the 2,000 to 5,000, we are definitely into yield loss territory. On some soils, I've lost as much as 50% of the yield due to cyst, especially when we're up in this level here of the high moderate. And then 5,000 or over, it's definitely to rotate to a non-host crop for 2 to 3 years. So kind of keep these numbers, the 40, 200, 2,000, 5,000, in your mind as I scroll through the next few slides. Okay. One of the easiest way to manage cyst is with crop rotation. And so if we are at relatively low levels of cyst, if we start at 50 and we plant a susceptible soybean, we'll usually typically go up tenfold in numbers. So the next year you would sample at the end of the season and you would have 500. The next year you would go down to corn, and that would decrease your levels by half. And then the following year, if you planted as another susceptible soybean, then you're up around 2,500, and then you're at the yield loss level. If you are in a resistant soybean rotation, alternating between susceptible and resistance, you kind of get this jagged, very slow increase over time. Sometimes with resistant soybeans, you'll actually see a decrease. We've measured that before. But if you're in a 3-crop rotation of soy, wheat, corn, soy, you're going to find that you're just going to constantly just keep at low levels for probably almost 20 years. So that's under low levels. When we start off in high levels, say this field started off with, you get the egg count back from your soil test and it's at 2,500, it's a different situation. So in this case, and this actually was in a field in Ohio, they planted a resistant bean and it stayed straight, but then they continued with the same cultivar. The pathogen adapted and it skyrocketed, just as if it was susceptible. If we planted the soybean corn rotation under these high levels, you can see at year 3, you are already in a very high level of cyst nematode. And so you definitely are in yield loss territory here. And again, if we're at even a high level of cyst and we're at our 3-crop rotation in the blue, only 1 year do we get a little bit out of it, and then you'll just kind of continue back down again. So, it's just a numbers game. A susceptible soybean, you'll increase population levels by 10-fold. With corn and wheat, you'll decrease them by 50%. And so those are general rules of thumb when you look at the whole population over your whole field. So what are the results of the survey that we've just done? So when we look at these classifications of the different population levels here, are none detected, 200, 2,000, 5,000, and too many. We had over 566 soil samples sent in. The worrisome number is this 10% and this 6% of the total numbers processed. And this tended to be the same percentage in each of the different years of the study. So I think this is a good general measure for the state. We're kind of in trouble here. 33, almost 34% of our soil samples from the 566 fields were in the range where we would be able to begin to measure yield loss. And so that's the first concerning thing. And so when we look at this from a pie chart standpoint, we still have a significant number that are none, which is this blue area, but it's not half anymore. It's become less than half of the total numbers that we sampled. Here's our hot spots, almost a third. And then we've got our 200 to 2,000, and these can go either way. A lot more are in this trace level that more and more fields have got cyst nematode in them. When we look at the number of counties, this is a slide that's up on our SCN u.osu.edu site. When we started the survey, we had this many counties where fields had been detected that had over 2,000 eggs per cup of soil. And now when you look at this, we've now added about 6 counties, Ashtabula, which is surprising, Auglaize, Dark, Lucas, Miami, Pike, Preble, and Ross. And then we've also added another county to the list where we now have SCN detected. So soybean cyst nematode was found in almost 60% of the fields sampled and processed. I think the last tabulation I did this was in December 2020. We've had a few more submitted since then, and we have very damaging levels in 10% of these fields. So obviously the first thing we're going to tell those folks is to start rotating crops, right? The next thing we're going to ask out of this survey was, is the 88-78 source of resistance still effective to our Ohio populations? So when we go back in our data, 96% of the populations, this symbol size goes the wrong direction. I'm going to correct this right now. The beauty of doing this on your right at the same time. 96% of the populations were this source of resistance was effective, and we measure this by the percentage of reproduction. Today, of the populations we tested, 86% of those had greater than 10% of the reproduction. So in this 20-year, 24-year time frame, we've seen this slow adaptation. Now, the good news is that we are not seeing a total collapse of the resistance. It's not like a Phytophthora resistance gene. This is a QTL, and there's many of them. They tend to act like a major QTL. So we do see an adaptation. So this is going to be a little bit different. And let me go into this a little bit deeper. So to put this in perspective, let's really dive into how is resistant measured. So here's our plant, our 88788, and here is our susceptible. And all these little bumps here are cyst females. And they're filled with anywhere from 200 to 250 eggs. So first, I think you can see if you had this many roots with this many cyst females on it, you can see why we get this exponential increase when we plant a susceptible variety, almost tenfold. But under the resistant, we don't. And so what this is, they do the math here. So this diagram here is representative of field A. And with field A, we have 120 females on the susceptible. There were only 5 on the resistance. And so that has a female index of 4.1 or a reproduction factor of less than 5%. Okay, actually 4.1%. When we go to field B, we have our susceptible and we have our resistant. And when we do the math, we have an 11.1%. All right, it's above the 10%, but it's still going to be effective and still be very good for your fields. When we look at field C, however, we now have 135, we have 40 cyst females on the resistant, and now we're up to a 29.6. This is where we start getting into trouble. Let me share some data that I received from my colleagues at Iowa State. So this is Greg Tilka's data. He runs a field evaluation every year of cultivars with the 88 source of resistance. And in these fields, he goes and he measures the female index at the end of every season. And you can see from 2000, it was very good. But then by the end of 2017, you can see that he's about 50% of the reproduction on the 88788 compared to that susceptible control. So even he's begun to measure this in the field. What does that cost us in yield though? So he had a student do this work. So when the reproduction factor was under this 10% line, you really can't measure a difference. Many of them were down here in that less than 5% range. But when we're up here in the 29 and 35% reproduction, you are now losing up to 14 bushels an acre. Okay, so if you have got a field where you know your cyst levels are high, you know that they're you're just not getting the yields that you're getting, this could be playing a role in this. It's not going to be an immediate collapse. We've had fields that have immediate collapse and then the yields are down around the 10 or 12. We're going to see a slow decline in yield, or where the new cultivars should be yielding 70, 80, 90 bushels. But some of these fields are just not moving out of that. And this could be one of the pests that's contributing to this. We then went with this data and we asked what was the reproduction factor on 88788 and also the cultivar Peking on these Ohio samples that had these high numbers. And these were from the samples that were submitted to us in 2018, 2019. And I think we're about ready to start on some of the 2020 samples we received. So this is a very intensive greenhouse test. It takes a minimum of 60 days and we can only run so many at a time. So it's been a bit a challenge. So I got to give a shout out to Zach and Tom, the two folks that have been really helping with this. But we've run 61 samples to date. And so we can see with a Peking, which is the deeper scarlet bar, A large proportion, almost half of the samples we've tested have very good resistance. The Peking resistance is holding up. However, when we look at the 88788, we can see that we're well above that 30% that I showed you. We have a couple samples that are over 60% reproduction on the 88788 relative to the susceptible. So we can see this shift begin to happen in real time as we go through here. Probably if reproduction is in this range, we're not going to be able to measure in the field, but clearly we're going to be able to measure yield loss when we're up at this level, if this continues to move forward. What are we going to do with this data? For you as a farmer, you're still going to buy varieties with the 88788. If you're up in here, we're probably going to tell you to switch to try to find Peking. But really, this information is critical for the companies, for the seed companies and the breeding companies, that they've got to get moving on finding other sources and incorporating different resistance to this pest. So that's the benefit of this research for the long term. Where did we detect greater than 20% reproduction? I went up to 20% because I think that's where things get more interesting. There are fluctuations. And so you can see where with the stars, that this is really spread out all over the state. And really these are about wherever we sampled in every county, 1 to 2 samples per county. So we are trying to fill in to find samples that we have for each of these, the rest of them, but I would dare say that this is present throughout the state. I think that would be my hypothesis as we begin to fill in. Peking, we do have a few spots where we're beginning to see this greater than 20% reproduction, but they are far and fewer between. So for cyst nematode, it's an integrated management plan, really. I mean, the ideal thing is just to keep the numbers low. Then we don't even have to worry about what resistance to plant, right? Because you're not going to see the damaging levels if you're already below that, 1,000 to 200 level. So when we do have it, we want to continue to use and continue to rotate our varieties with the 88788. If you're in that high level, you do want to seek other sources of resistance, add other types of resistance when they become available. We do want to use nematode protectant seed treatments. There's some data that supports that maybe this reduces the number of full generations we get in a season. I think the data's, the jury's still out on that, but that's one of the new suggestions that we've been hearing. Cover crops, summer host. So I don't like to say anything negative about cover crops, but I think in these high cyst fields, we do have to take a look at this. So as like clover, bird's foot trefoil, some of the beans, cowpea, lupin, sweet clover, the cyst actually reproduce just as well on these as they do soybeans. The other thing that cyst nematode likes are weeds. So chickweed, henbit, purple dead nettle has become one of our signals. Sometimes a good purple dead nettle field could also be a good cyst field. Wild mustard, and of course field pennycress as well. So if you haven't sampled and now you're beginning to think, oh, there's that field that I have that just isn't moving up in that 70, 80 bushels per acre yield, maybe I need to take a cyst. Here's how you do it. If you've never taken a cyst sample, I would just target those low areas when you look at your yield maps. Target those spots first, because if that's where you're going to find it. Otherwise, just however you take your fertility sample is the same thing. We've done some work with some of the automated soil samplers. They work just fine because with cyst, it's in that top 6 to 8 inches. And so it's in that root zone area. So if you're sampling for fertility, cysts are in that same area. And we need about 1 gallon of soil for every 20 acres. If you're doing a diagnostic spot, make sure that we have a good 3 cups of soil. So if we do find it, we can run the SCN type test later on. So there's tons of information. So they have, many of my colleagues this season with COVID actually made quite a number of videos related to sampling and the biology of cysts. There's some nice pieces through the SCN Coalition, some farmer stories. I think I've shown them to you in some other presentations. So while you're home, here's the link. There's lots of information there. Resistance, rotate the varieties and within the 88788, because what's cool about the 88788 is it has so many genes for resistance that one variety doesn't hold them all. And so by switching up the genes from the source or switching varieties that have different suites of genes, you can actually continue to control this pest for the time being. And if, of course, we're going to report that you've got some cyst populations that have adapted to the 88788, then by all means begin to look for Peking. Okay. There are SCN testing labs available, including our own diagnostic clinic, Spectrum and Brookside. Everybody is now moving to egg count, so there's other cyst testing labs now. There's been several companies responded to this as a need, so they're all very good. It is a random, because you're picking up a soil and you're going to try to make sure you get that cyst out of it. So there is going to be fluctuation among tests for the labs. So with that, I see we have 6 questions. So Mary, why don't I pause here and why don't we take a look and see what the questions are for cyst?
All right, I'll go ahead and handle those for you, Anne.
Oh, thanks, Amanda.
No problem. So what % of soybean varieties are resistant to SCN?
Right now, most of them. I think the companies, because it's so hard to breed for, you're going to find that most of them have some resistance to this. They might not have all the genes from 8878, but they have some. And so when you're looking at that score, if it says R3, that actually comes from the 88788 source of resistance.
Okay, thank you. And what is your opinion on the efficacy of seed treatments that contain nematicide that claim suppression against SCN?
I think when I alluded to the seed treatments, that's where this is coming from. I think you might get that first generation, you know, that it's not going to taste good, right? And we do know that with the Allevio, there was some data, that there was some decline in cyst in those plots over time. So that would attribute to this, just not that first generation getting started. So I think there's more work to be done on that. It's very tedious, hard work, and we need a couple good graduate students on that are very particular to get that to work.
Okay, and then the next question is there a list of cover crops that are SCN hosts?
Yes, there are. It's in our fact sheet that we have at the u.osu.edu site. If you just type in soybean cyst nematode, there's a whole table of those that are there. And this is actually work that goes way back by several people at Ohio State early on.
All right. That's, I think, all I see for now.
Okay. All right, let me proceed then into, I almost apologize for this slide. This brings back horror stories, right, of some of our springs, but we have been in some very wet springs, and they've definitely met their challenges. And so what I'm going to move into now are the root rots, some work that we've been doing with the water molds, and kind of finish up with some of the seed treatments. So this is very classic of our conditions, and these saturated soil conditions really favor a lot of these seedling diseases that are caused by water molds. And the reason is under these flooded soil conditions, which are represented here in this blue saturated soil, right? These types of pathogens form these beautiful sporangia, which form a swimming spore. And what's cool from a biologist's point of view is these are actually attracted to young roots. And then they will then germinate and grow and infect the plants. So our most common in Ohio, what we're best known for is Phytophthora. It's been here since the late 40s, early 50s. We've just finished a new population survey, so I'm going to jump into that data next. So we had isolates, I think we had well over 200 here from Kentucky, Ohio, Indiana, and Illinois. And in Ohio, this color code actually stands for the number of RPS genes, the number of resistance genes that are not effective, that don't work. And so you can see in this historic Phytophthora territory that we are already up to 8 or 9 different resistance genes that no longer work. Wood County, we're up to over 12. So none of the resistance genes are effective to the isolate that we tested. And so for Ohio, what we've been saying for a long time is don't focus on your cultivar selection on the RPS gene as much anymore, is really focus on the other resistance that's effective, the quantitative resistance, the partial resistance, some companies call it tolerance. Here's another way to look at this. This was funded through North Central Soybean Research Program Checkoff and Ohio Soybean Council. But from the percentage of samples, Ohio is the blue bar. I don't know why I did Illinois in red, but anyways, we can see here that 1A is not effective, 1B is not effective, because this is the percent of the genes, the percent of the isolates that cause disease on that. So we're almost, sorry about that, we're almost at 100%. So this is why we're really focused on this stage. If we have high partial resistance, we never see the stem rot phase. And so if there's no stem rot, then we don't have any late to mid-season stand loss. And that's really our goal for this disease, to get through the season. The other pathogens that have the same basic disease cycle are all the pythiums. And there's probably over 30 that are pathogens of soybean and corn. Within Ohio, our first student that began to look at this was Kirk Broders. He's now at USDA ARS in Illinois working. So we can see that each of these bars is a different species. And here it's whether they're a pathogen of corn and soybean. So some favor corn more than soybean. We then went and asked, because I often get told, well, pythium's only a problem when the soil temperatures are cold. So we took soybeans and we planted them in soil temperatures, and we flooded the soil at 2 temperatures, 15 and 25, or just about 60 and 77 degrees Fahrenheit. And again, we're pulling out a large number of species. And what I found fascinating, each species is a different color here. The color density, the number of colors is quite different, both across these 5 fields, which were managed differently, but also between temperatures. So there is plenty of pythiums out there to attack our corn and soybean seedlings in any given year. It just needs to be wet. And I think that's the theme of this. A lot of work to kind of come to that conclusion. The other thing we found is that there's a different complex or a different community of these pythiums in all of these different fields. So each of these squares indicates a different field across the state. When we ran this study, I think these are about 14 locations across the state that we went in and we took soybean seedlings and we did the sequenced up the microbiome on those to see who was present. And you can see there's just a different group in every single field. What was interesting was we sampled 2 fields at 2 different time points. So this is Defiance at planting, or excuse me, at 14 days after planting we sampled. And then here's at 25 days after planting. We have 2 totally different communities of pythium that are popping up in this. So it just really needs to be wet. The age of the plant, the temperature of the plant, how that field was managed are all going to play a role. But the bottom line is we just have to manage this as a community and not as a single pathogen. To manage as a community, though, is going to be a little bit difficult. The first thing I do is I send the students to a population and say map resistance. So this is a circus plot where every one of the 20 chromosomes is around the circle. And the bars that you see are locations for different Pythium species, including Phytophthora. And you can see resistance is scattered all over the place. There's no one locus that's going to confer resistance to all of these. So this is from the seedling stage. This is going to be a challenge for breeders, which again is going to put us more emphasis, I believe, on using seed treatments. So currently, there are on the market metalaxyl mefenoxam, which has been on the market for quite a while. The strobilurins, ethoboxam from Valent, we contributed a lot to the primary data to get that labeled. Oxothiopropylene is another one. We've submitted a paper related with this. And then there's going to be another one that's pending EPA approval that we've contributed data to as well. So Ohio State has contributed to getting 3 seed treatments labeled with efficacy towards these water molds. Now the strobilurins are kind of interesting. I'm going to skip through this to save time for mode of action, but it's in the mitochondrion and it's an energy savings, and it's a single site. So we do see resistance develop. Same thing with metalaxyl. It's in the single site mode of action for the biosynthesis of RNA. Again, we see resistance develop in this pathogen population. And then for a lot of the years that we've done this, and I just pulled this data because it was nice and clean. You know, when we add under low rainfall and really high rainfall that we have, we can see a little bit better difference. But when we do add the seed treatment, we are definitely significantly above the base seed treatment. So it doesn't always work. It's really dependent on how much rainfall we get and across 2 years. So very consistent data there. Okay. The one thing I wanted to bring to everybody's attention was some data that we developed. Oh, it's been 10 years ago now, but it's going to come into line again this year. We had a lot of seedling blight the year after the 2009 outbreak of corn ear rot back in 2009. So there are parts of the state where we're seeing the same thing. One thing to note, Captan isn't used anymore on soybeans, but if you are in one of these pocket areas where you do have high Fusarium graminearum on your corn, you're going to want to up your rate of Maxim or Fludioxonil up to the high rate. When we're at the low rate, we're not getting total control. You're going to see these reddish lesions form. You might think it's Rhizoctonia, but in fact, Fusarium graminearum does attack soybeans. Here's some other data that we have from another student. We looked at seed-borne, and so in this case, really the Fludioxonil low, we're still getting some Fusarium come through, but when we use the Fludioxonil high, it's clean. The other thing is, if you don't want to go with a high rate of Fludioxonil, tebuconazole plus Fludioxonil at the low rate actually gives very nice control too. So for those of you that are in this heavy corn, ear rot, stalk rot locations, and you're going in with soybeans, it's something to kind of adjust in your seed treatment window. Fusarium graminearum gave some control by the strobilurins, but it's really not enough to get too excited about. So with this, seed treatment packages look like they've got everything but the kitchen sink, right? So with these, we've got Phytophthora and Pythiums, the water molds when soils are saturated. We've got a range of rates among these different things. They are all good. With the Syngenta, the azoxystrobin, the pyraclostrobin, and trifloxystrobin, we have a range. And then we've got no control for the Rhizoctonia and Fusarium. When we look at full combination products, though, instead of these products singly, everything becomes good across all the different organisms. And so that's why. I mean, we're controlling things that are only attacking plants when it's wet, actually saturated soils. Those organisms swim to the roots. They require, they just have a different biology than the true fungi like the Phomopsis, Fusarium, and Rhizoctonia. And so that's why we're using different seed treatments for those products. Ultimately, though, with this, the risk becomes down to what's the weather going to be. So for seed treatments, do you have a history of replanting? Do you have poor drainage? Are you in a no-till, reduced till? Because in those, we know that the inoculum density is very high in that upper levels of soil. And do you have a history of just poor stand? And it's not just from crusting. You know that there's something else going on. And when you walk through you see these little dead soldiers, little brown decaying seedlings. These are the places where a seed treatment's going to make the difference for you. And again, a reminder with Phytophthora, there is resistance for Phytophthora. Just make sure it's the quantitative resistance. This is a line I use for a lot of demonstrations. It only has one gene, one of the RPS genes, and it is not effective wherever I plant this. So it's a good marker for my team for the end of the season. Okay. And with that, I'm going to wrap up here because I think I'm out of time and want to leave time for questions. So I want to thank everybody. Hopefully you're all staying safe. And this is Zach, who's counting more nematodes. All those pink things are the nematodes that he's counted, and all of our funding through the checkoff for the coalition, the Phytophthora, and the companies for the seed treatment to be able to run those seed treatment tests. Okay, Amanda.
Yeah, one question. We are confirming much higher numbers of both northern and southern stem canker in Pennsylvania and New York over the last 3 to 4 years, often confused with PRR. What are you seeing with the incidence of stem canker in this area?
That's a good question. So not so much last year, but definitely the 3 years before. And northern and southern stem canker tended to be on the later maturing varieties, late 3s. I think there was 1, 3, 3, and definitely in the 4s. And so especially with southern stem canker. So with southern stem canker, you'll drive up to a field and it looks like it's dying. And it looks like charcoal rot. It can even look like sclerotinia sometimes. But when you walk into the field, you'll see red lesions scattered up and down the stem. And when you cut them open, it'll look like brown stem rot, really. Only the whole area will be necrotic. Really, and sometimes it's really red. I thought that was really fascinating. So with northern stem canker, it has the same chocolate brown canker that Phytophthora does, but it's limited to that 3rd or 4th node. You can actually, at the bottom of the plant, you can actually have a shoot that's really healthy come out to the side. Both of these diseases will tend to come in just as that plant is really filling out the beans, about that R6, between R5 and R7. Same time we see sclerotinia. So that's a time to watch for it. I think the companies have kind of caught their eye because it was the whole field in several cases, really creating this issue. And I think we shouldn't see those varieties anymore. And so just to keep an eye on for it. But if you've had it, if you're one of the places where we diagnosed it, the recommendation is wait a couple years, till that under, so we don't have that residue surviving. and then really watch the resistance ratings. I mean, both of those diseases are well controlled with resistance.